Hey, we're going to do photosynthesis, respiration, kitchen biology, right? We're going to make it short and sweet. Mr. Trevelyan is tired and a little bit cranky. Hey, this is the equation for respiration. Tongue in cheek. What do you do before the test? You take a deep breath. What's going to go in is glucose and oxygen. What comes out is carbon dioxide and water. If I know that, then I also know photosynthesis. Because if you'll notice, their reactants are our products are their reactants. A symbiotic relationship called mutualism. Both organisms benefit, but on a big global scale, right? So we're going to take in glucose and oxygen, give off carbon dioxide and water. I didn't have a bottle large enough to fit Gail in, Mrs. Trevelyan, so I'm going to use yeast. Yeast turns glucose into carbon dioxide and water with the help of oxygen. Yeast is a eukaryotic, membrane-bound organelles, and one of those membrane-bound organelles are mitochondria. If it has mitochondria, it does this process called cellular respiration. This is the chemical formula I had you memorize, C6H12O6. And this is the breath we take in every day. I will strangle you if you don't get that right on the test. Then you'll know. And we're going to turn carbon dioxide into water. We're going to use yeast because we still got packets of yeast and we're dealing with a pandemic. What yeast are what we call facultative aerobes. What I mean by that is they can take oxygen or they can leave it. They can do fermentation without oxygen, but they can also do cellular respiration. They can do this process up here. Now, the couple of byproducts you'll realize from yeast are alcohol and this gas called carbon dioxide, right? So today, we're going to let them do some cellular respiration. Eventually, they're going to be doing fermentation, or what we call anaerobic respiration. But we've got some yeast in our little bottle here. I don't know what this is. Sparkling ice. Fancy. And what I put in there was a thing of yeast. I think you still might have one in your kits. And then what I'm going to do is give it some food. C6H12O6. Actually, it's a disaccharide, but you get the idea. When it breaks down, it's going to be C6H12O6. What you're going to turn that into is carbon dioxide. I am assuming, I'll be honest with you, I don't know the truth about this, but I am assuming we're going to still be doing aerobic respiration, cellular respiration, with all this oxygen up here. But eventually, I got the balloon on top. I'm assuming it's eventually going to be doing fermentation without oxygen. And Mrs. Trevelyan gives me a confused look. What's up?
So if the balloon, I don't know.
Not sure. Mrs. Trevelyan asked good questions.
C6H12O6.
It's going in, baby. We're going to go crazy here, too. 1.
Oh, it's clogged.
No, it's not clogged. Maybe a little clogged. We'll put a little water in there eventually.
You could squish
Then we're going
those.
to put 2. Oh, this is live TV.
There it goes.
And give it a little more for good measure. More glucose means more carbon dioxide. More carbon dioxide means
More fermentation.
more fermentation or respiration.
Oh.
I'm assuming at first it'll be, well, let's mix that up a little bit. Let's give it the oxygen it wants. It so desires.
Oh.
Yeah, hey.
Whoopa!
Whoopa. You might want to hold that bottle, Mr. Trevelyan. Yesterday, I spilled it all over myself. All right, we'll come back to that puppy. This thing should fill with carbon dioxide. I need a way to quantify our data, right? Can we measure the size of the balloon, the diameter of the balloon as that gas builds up? I'm not sure why, but at first it usually sucks in. Anyway, we'll come back to that. How are we doing time-wise? We should be at 5 minutes because I got to do photosynthesis.
440.
You're amongst professionals. All right, that's going to sit over here. Hopefully, by the time we're done, we'll see a little bit of air in there. Oh, look it! I see bubbles. That's oxygen bubbles. Carbon dioxide bubbles. Not oxygen bubbles. Let's make oxygen bubbles next. Mrs. Trevelyan, we are going to do photosynthesis next. Notice,
It's true.
over here! Notice that we're making glucose this time. This is anaerobic. We're gluing the vase together. Some of you didn't like that analogy, but it's the one I like. We're gluing it together, putting it back on the table. There's going to be more energy in this glucose than there was in the carbon dioxide and water. Again, the sun's beating down, right? We're going to add 2 drops of liquid soap. Pretend there's still some liquid soap in here. And 2 pinches of baking soda. This is going to be the carbon dioxide supply, right? When it goes in water, it produces CO2. And we're going to use that CO2 so that plants can do photosynthesis. They're going to take the carbon dioxide and water and they're going to make glucose. And more importantly, at least for our experiment, they're going to produce oxygen. And that's what's going to make these little leaves float. Okay? We're going to have 1 dish beaker over there without baking soda. That'll be our negative control. We expect no disc to move at all. And then the plant disc, we never did decide. Is that?
That's right.
All right. The plant disc fills with oxygen as photosynthesis occurs. So in this case, we're going to take these little, if you had a hole punch, but otherwise I figured out the straw works okay. You're going to make these little discs, right? See that one, Mrs. Straw? Like that?
Look at how cute.
Yeah, look how cute. We're going to put them in with the soap and the baking soda. Remember, the baking soda is the carbon dioxide. The soap is going to take this hydrophobic material off the leaves. There should be a lot more water in there. Okay? Now, the next, I got to speed it up here. We're at 6 minutes.
Yeah, 6:51.
Man, I sucked up some of the baking soap water and a little bit of soap with those discs in there. How well can you see those discs?
You can see them, right?
Okay. Wow, let's hope so. Anyway, I put just a little water in there, and now what I'm going to do is create a vacuum. I'm putting my thumb over the syringe, and I'm pulling out. Oh, that didn't go as well. Anyways, shoot. All right, let's assume, though, there's still some water in there. Now the discs have all gone to the bottom. The reason they've gone to the bottom is I took all the air out of the leaves. The only thing that's going to put air back into those leaves is photosynthesis. What air is going to go into the leaves, Mrs. Trevelyan?
Oxygen.
Oxygen. Oxygen is correct. Now, you're going to set up your syringe with your little disc. You got this. I even brought in some spinach. You can use a leaf outside. I don't think it matters. As long as it's a living leaf, it should work. Right? It'll still be doing photosynthesis. It'll take in carbon dioxide through those stomata, and out will come oxygen. And we'll know that because the disc will begin to float again. So if you look over here, we can do several things with this experiment. Here's my negative control, just water, and these little discs in there should be at the bottom, right? They are not. But that's the point of a negative control. It's a comparison group. These guys might be floating for photosynthesis. But these guys, if they're floating, they're not doing photosynthesis. They have no evidence of carbon dioxide. Agreed? They need carbon dioxide to make photosynthesis. And photosynthesis produces oxygen. And that's why they float to the top. Now, easy way to quantify our data would just be time. How long does it take for those discs to go from the bottom to the top? Assuming that every disc that gets to the top is now producing oxygen. 8 minutes?
9.
9. Quickly, look at this. Predict, make an if and then hypothesis. I put blue, this is just so cool, and red and green to filter out the other lights. So this guy's getting blue light, this guy's getting red light, that getting green light. Which light do you think plants do photosynthesis best at?
Green.
Green?
That's my guess.
Well, green now is the color of the leaves. They reflect it. Want to change your mind?
Oh, my favorite color is red, so I'll go.
I am seeing very well. Here, let's show you. How much time we got?
It's 5 seconds.
5 seconds. All right. we'll start to see if we can see a little bit of air coming into this thing. I put a lot of sugar in there, but very soon we should see expansion. I think when I played with it earlier was, I don't know, at least a half hour before I got some air in there. And I had my freshman mixing up the bottle for me. Oh, look it. All right, this is Biology Bites. Go try these labs. We're safety. Oh, nice. We're safety goggles. Bye.